All right, day 2, exceptions to Mendel. Let me make sure I get my little pointer here. All right, so we're going to start off with sex-linked traits. Sex-linked traits are focused on our sex chromosomes. So thinking about our karyotype, sex chromosomes are those 2 chromosomes that determine our sex. Are we male or are we female? If we're female in humans, we have 2 X's. If we're male in humans, we have 1 X and 1 Y. Sex chromosomes are very specific and special. They determine our sex. All of the other ones are called autosomal chromosomes. So in humans, the other 22 pairs, or 44 other individual chromosomes, are called autosomal chromosomes. They are not as special as those sex chromosomes. They still are important. They give us our other genes and alleles and attributes and all of that good stuff. But there is a difference between those autosomal ones and our sex chromosomes. That's why they have different names. So sex-linked traits, they were first discovered by this guy down here in the bottom right corner. His name is T.H. Morgan. He did most of his research at Columbia University. If you're interested in Columbia University, hey, little factoid. He worked with Drosophila, also known as fruit flies. Drosophila is the sort of scientific genus species name, but they are fantastic subjects for genetic research. They are prolific. There's a lot of them. When they do breed, they breed a lot. There aren't just like humans where when we reproduce, there's only 1 baby. Fruit flies, there's hundreds for each generation. Prolific. Also, they have very, very short windows of life so that they reproduce and produce generations very quickly. They have 2 weeks for each generation. That's super duper fast. For humans, it's like decades between generations. Super short lifespans means super quick reproduction rates, very short time for new generations. That means we can study many, many generations in a short period of time. Another reason why fruit flies are so great is that they only have 4 pairs of chromosomes. That's it. Humans have 23 pairs. Fruit flies only have 4. So they're fairly simple when we think about it from a genetic standpoint. 8 individual chromosomes. Very simple creatures. The other and last best point about these fruit flies, their sex chromosomes are the same as ours. 2 X's mean it's a female, an X and a Y means it's male. Down here at the bottom left, we have a picture of 2 different fruit flies here. One of them has red eyes, one of them has white eyes. That'll become important when we look at our next slide. Or sorry, next slide. So here, again, I want to point out the difference between our chromosomes. Here is a human karyotype of a male. How do we know it's male? Well, we look at our sex chromosomes here. We have 1 X and 1 Y. Please notice the difference. They're not as homologous as our other autosomal chromosomes. So all of these guys up here, 1 through 22, are autosomal chromosomes. Notice how these autosomal chromosomes are generally the same shape and same size. They are homologous. In a male human, excuse that card, here are our sex chromosomes. They are not homologous. They are different shapes and different sizes. In females, they are homologous. They are the same shape and the same size. We have 2 of the same. We have X and X. But males are not homologous. They are just, they're still considered a pair because we need 2. We are diploid. And so this little slide here is just showing us the difference. Autosomal, again, means all of these chromosomes that are not sex chromosomes. All right, here we go. Here's our fruit flies. So we're going to look at Morgan's sort of experiment here, go along with him in his journey to discovering this sex linkage. So on the left, we have a true breeding red-eyed female. I know, pink and blue, kind of gross, going back to, sort of 50s assignment of colors with genders, but it's what we know. It makes it easy to just quickly look at and identify. So here's a female. I colored her pink just because, generally speaking, unfortunately, females are associated with pink. And she has red eyes. Over here, we have our true breeding male. Again, blue just because our society associates blue with males. And he has white eyes. They reproduce, and lo and behold, we have 100% red eye offspring. This tells us that red eyes are dominant. If we have two true breeding, Mendel tells us all of their offspring will be heterozygous. If it's heterozygous, it will display that dominant trait. The dominant trait now we can see is red eyes. So we have some females, some males. Both of them have red eyes, 100%. Checks out. Heterozygotes have that dominant trait present. All right, so now what happens if we take that F1 generation, our 100% red eye offspring, and breed them together? Whoa, this is new. This now F2 generation, we're seeing all of the females still have only red eyes, but the males are 50-50 red eye to white eye. Interesting. This is a new phenomena, and Morgan was like, this is weird. This doesn't align with Mendel's work. Maybe I'm onto something. So this is what it should look like if we're doing Mendel genetics here. We have our dominant red eye, we have our recessive white eye, we put it in a Punnett square, we get heterozygotes. That part checks out, great. Then we take two of our heterozygotes that were part of our F1 generation, breed them together. And what we should see is regardless, so these are brown flies, because regardless of gender, it shouldn't matter what color their eyes are. Regardless of gender, we should have 3/4 or 75% with red eyes and 25% or 1/4 with white eyes, but that's not what he found. He found that 100% of the females, only the females, 100% had red eyes, and then the males were 50-50. So this doesn't align with Mendelian genetics. Well, now we have to take a little bit closer look at our sex chromosomes. So in humans and other mammals, there are two sex chromosomes. We know that. X and Y. Females have two X's. Males have the XY. Because females have two X's, we have homologous sex chromosomes. We have a sort of redundancy or protection from recessive alleles. We have a greater chance of having that dominant trait because we have two X's. Males, on the other hand, only have one X. So if there is a trait associated with that X gene, you're going to be stuck with it. Looking at our chromosomes here on the left, our two X's, as you can see, they're pretty much the same size and shape. The males, we have one X and one Y. That Y is very itty bitty compared to our X chromosome here. They are different sizes, different shapes. When we plug in sex chromosomes into a Punnett square, would you look at that? 50-50. Thanks, Mendel, for making these Punnett squares. We can see that whenever two sexes sexually reproduce, we're going to have a 50% chance for each. Possibly 50% female, possibly 50% male. So that's why when we have our gender reveal parties or whatever, there's a 50% chance that it could be a girl, 50% chance it could be a boy. We get an X from mom and an X from dad 2 times, and we get an X from mom and a Y from dad 2 times, 50-50 chance. So keeping in mind how our chromosomes and our sex chromosomes are, what happens if we attach the alleles, and we attach that trait for eye color to our X chromosome? If you remember from this slide, look how much bigger these X chromosomes are than this little Y chromosome. That means if there's a trait attached to one of our sex chromosomes, chances are pretty good it'll be on that X one. There's more room for more traits. So usually when we talk about sex linkage, we're talking about it's attached to that X gene, or sorry, that X chromosome. So here we go. Here's our parent generation. We have a true breeding female. She's female. We have 2 X's. She's true breeding, meaning she has 2 dominant alleles for red eyes. So capital R, capital R. We're using superscripts here as a sort of note on that sex chromosome. Again, sex chromosomes are more important and special compared to all of our autosomal chromosomes. So we want to make sure we know that we have sex chromosomes here. And then the superscript is where we attach that allele to show our genotype. So we have a true breeding female here. We have a true breeding recessive male here. Again, it's attached to the X chromosome. He only has one of those, and he gets a little R on that X chromosome. Put the male on the top, put the female on the side, fill in our Punnett square. We get an X with a big R from the mom, an X with a little r from the dad. Next square, X with a big R from mom, a Y from dad. So these two will have red eyes. We have at least one big R. That will mean it's dominant for red eyes. Third square here, we have an X with a big R from mom, an X with a little r from dad. Again, heterozygous, that red eye is dominant. Our fly will have red eyes. And our last square here, X with big R from mom, Y from dad, dominant R, we're going to have 100% red eyes. 100% red eyes. That checks out. We have at least one capital R for each. Makes sense. Now we're going to take our F1 generation, breed them together. We have a heterozygous female and a dominant male. That's all we get. We get heterozygous females, dominant male. Put the male across the top, put the female down the side. First fly here, X with a big R from mom, X with a big R from dad. Homozygous dominant, it's going to have red eyes. Next box, X with a big R from mom, Y from dad. Dominant for red eyes, going to have a red eye. Third box. Remember, best practice says capital comes first. So we're going to take the X with a big R from dad, X with a little r from mom. Heterozygous, dominant for red, going to have red eyes. And lastly, we have our last fly here, X with a little r from mom and a Y from dad. Because it has a little r and only one X chromosome, it doesn't sort of get that quote unquote protection of the redundancy of another X where it could possibly have a dominant trait attached. Now we have our recessive male. Looking at our ratios here, 100% of our females have red eyes. That looks like what Morgan found the first time. And now it's making sense. 50% of our males have red eyes. 50% of our males have white eyes. Again, that allele for white eyes is attached to this X chromosome. This one has an allele for red eyes attached to the X chromosome. They don't have that redundancy of a second X to have another opportunity for making a heterozygote or a homozygous dominant. So for the males, it's either or. You only get one allele for your eye color. It's either going to be red or it's going to be white. So like I mentioned earlier, sex-linked almost always means X-linked. It's going to be on that X chromosome. Looking at the size difference, X chromosomes are much bigger. There's more room for all of these different genes than the little teeny itty bitty Y chromosome. Here are a list of a bunch, not all, of some of the traits we found associated on this X chromosome. More than 60 diseases have been traced to genes on the X chromosome. Some of them have interesting names. I haven't heard of some of these. Some you may have heard of. Down here we got some hemophilia. If you want to take a look, read through these on your own. Awesome. We're going to move on. So here is something we call a pedigree. We're going to look a little bit deeper in that in a few slides here. But what a pedigree is it's kind of like a family tree. We have parents up here at the top, then they have all their subsequent generations, kids, grandkids, great-grandkids, nieces, nephews. We got uncles and aunts and all of those cousins and stuff. So it's kind of like a family tree, except it's sort of tracing and tracking one trait. In this case, we're looking at hemophilia. Hemophilia is a disease that where your blood doesn't clot as quickly as it should. So when you get a cut, usually the bleeding stops in a couple of minutes at most. You know, oh no, I picked up a piece of paper wrong and I got sliced. It's going to bleed for a couple of seconds probably, and then it'll clot. but hemophiliacs will take much longer for their blood to clot. It ends up that Queen Victoria, I think this is her here in the middle, and all of her descendants, as you can see on this pedigree, are hemophiliacs that, you know, they could be present, might not be. It's attached to that X chromosome. So this pedigree is tracking that trait for hemophilia through all of these different relations. So let's look at hemophilia really quick as just another example of sex-linked traits. It is recessive. It is recessive, just like those white eyes on our Morgan's flies. So now we're going to do it with an H for hemophilia. Whenever you see a circle on a pedigree, that stands for a female. So here we have an X and an X. Whenever you see a square, that stands for a male, an X and a Y. So let's see what happens when we cross a heterozygous female with a normal or dominant male. Again, hemophilia is recessive, so we're going to need the little h's to have that hemophilia be expressed as a trait. Put the male across the top, put the female down the side. Here we go. We get an an X with a capital H from each parent. This one is homozygous dominant. It is a female. We have XX, so we put it in a circle. Over here, we get an X with a capital H from the female and a Y from the male. Put a square around it. It is male. It has an XY sex chromosome. Third box down here, we get again, best practices, capital comes first. Capital H on this X chromosome from the male, X with a little h from the female. It's got two X's. It's a female. We'll put it in a circle. And lastly, this guy over here gets an X from mom with a little h and a Y. Put him in a box because he's a male, but let's look at what happened here. Again, we have that case where it's a recessive trait. There isn't the redundancy in males to have, you know, another X possibly with a capital or dominant trait to kind of dominate over that recessive trait. So whatever you get is whatever you get for males here with hemophilia. You either get a capital or a lowercase. You don't get a second allele. Here's just on the right side here, we get a breakdown of how we can break up our chromosomes here with their H's or alleles attached for hemophilia, if you ever get confused. Now let's look at a different type of sex-related or influenced trait. So male pattern baldness is an autosomal trait, so it's found on those autosomal chromosomes, not sex chromosomes, the other kind, but it is influenced by sex hormones. Females tend to produce more estrogen and progesterone. Males tend to produce more testosterone. Both sexes have all of those hormones, they just produce them in different amounts. Age is also a factor. Male pattern baldness typically starts to show around 30 or older. Some people it's a little bit earlier, some people are a little bit later, but around that 30-year-old mark. It is dominant in males. So that's why here we have a genotype with a capital B. It doesn't matter what the second one is. It could be capital, it could be little, doesn't matter, it's dominant. But guess what? Females can also have pattern baldness. It's just much less common because it's recessive. We have to have those two little b's in order to have pattern baldness in females. Again, some of it has to do with those hormones that are produced by each sex. Some of it has to do with age, but it is influenced by those sex chromosomes because based on your sex chromosomes, you'll produce different levels of different hormones. All right, now let's look at our pedigrees here. This is looking at red-green color blindness, and it is an X-linked recessive trait. So just like hemophilia and just like those white-eyed fruit flies, it's going to be recessive and it's going to be linked with that X sex chromosome. As a reminder, the squares are males. So wherever you see a square on here, that is a male. Circles are females. Wherever you see a circle will be a female. If it is completely shaded, so full, in this case, blue, it will be expressing that recessive gene, in this case, the red-green color blindness. So whenever you see a fully shaded red or a fully shaded circle or square, they have that red-green color blindness, that recessive gene. If it's not shaded, if it's just blank and white, they don't have any of those genes. They're only dominant. They're good to go. They have full color vision. These half-shaded ones, whether it's on the right or the left, I don't know why they flip-flopped there. But if it's half shaded, that means that person is what's called a carrier. They carry the gene of this recessive trait, in this example, red-green color blindness, but they don't express it. So these are going to be our heterozygotes. They have the dominant trait, so it's not expressed for them, but they still have that recessive allele that they can carry with them and pass on potentially to their offspring. Lastly, I want to touch on environmental effects. Phenotypes can also sometimes be controlled by environment. Yes, we know we get a lot of our genes from our parents. Biologically, down in our DNA, we get alleles from our parents. But sometimes the environment can have a factor. Right here on the left, we have a flower called the hydrangea. Notice how they're both kind of in the purpley zone, but one is more blue and one is more pinky reddish color. That range is actually dependent on the soil and how acidic or basic it is, that pH of the soil. I don't remember off the top of my head which one is which one is more acidic, which one is more basic, but they're both kind of purpley, but one is more blue and one is more pink. Depending on the pH of the soil, you'll get a different color flower. Right here in the middle is something we're all familiar with as Southern Californians. We are constantly in sun. We know we get skin color from our parents, but sometimes hanging out at the beach, going sitting out by the pool, playing out in the sun during the summer, those sun rays, the UV from the sun, can make our skin a little bit darker than it would be if we weren't out in the sun. Another example of the environment being a factor in one of our phenotypes. Lastly, we have this cute little fox up here on the top right. Arctic foxes actually change their coat color depending on the temperature or season. In the winter, obviously, it's kind of snowy, it's cold, they're going to have a whiter coat to sort of blend in with their surroundings, with their environment. When things start to get a little bit warmer, the snow melts, we get more green and brown with the soil and the plants. Again, their coat will change colors to sort of help them camouflage and be successful as predators. An environmental effect, the temperature or season, affects and changes their coat color. So sometimes environment can be a factor. All right, questions, comments, concerns, clarifications? Do I need to go back for notes?